Okay, this is we actually like kind of summarized the ARTIC protocol. Before we've had online discussions with other labs who've been doing the same thing. So we also incorporated some of their suggestions. And fair enough, for the first two batch that of sequencing that we did, we've had good results. The protocol is working as expected. So I think I'm just going to highlight key important stuff, which might be confusing if you are just starting with SARS-CoV-2 sequencing using the ARTIC protocol. The first one is in terms of sample selection. So basically, based on the ARTIC protocol, you need to have a CT value of below 30. But that's not a blanket assurance wherein if you have a sample with CT below 30, you automatically get good results. That's not always the case. We've had cases wherein samples with a much lower CT, which means higher concentration of RNA, like 22 or 24 CT values, having much more less quality results compared to the ones with bordering 30 CT. So some samples bordering 30 can also be sequenced successfully. So we don't know yet what the exact reasons are, but we kind of hypothesize that this could be an issue of sample quality. So for instance, if your samples are being transported from a laboratory and along the transport route, there could be issues which could cause partial degradation of the samples, hence leading to a reduced quality of your sequencing data. But generally, we are in our lab we've used Allplex 2019 nCoV kit to retest all samples that are being submitted by COVID-19 laboratories, and there are instances wherein SARS-CoV-2 positive samples, after being submitted to us, are testing negative. We're not saying that the original diagnostics test was wrong. We're saying that along the way, there could have been issues with sample quality, and it reached in our lab, the viral RNA is already undetected. So to avoid, wasting precious sequencing slots, so we could we always ensure that the viral RNA is still detectable prior to starting library enrichment, sample enrichment, and library preparation. So the rest of these guidelines from the ARTIC protocol as is. So you do dilution depending on the CT range. Yeah, and sometimes when it's difficult really to. There are cases when the diagnostic test use more than one marker. Like for instance, the Allplex test uses 3 markers, like the N gene, the E gene, and the RDRP gene. So basically, as long as all of those 3 markers has CT value within a particular cutoff, like below 30, we usually test them. But we've already tested. We've also tried testing samples with CT values higher than 30, like 31, 32, 33, and we also get partial genomes, which some of which cannot be assigned a lineage. A few can also be assigned a lineage, but the quality is generally worse compared to samples with low CT values. So I think that's what we can say for a sample selection. For cDNA synthesis, we just follow the protocol as is. So just basically what was written in the ARTIC protocol. We did modifications in multiplex PCR because in our experience, if you look at the protocol, the reaction volume, like let's say a 25 UL reaction volume, and you only need like around at least 2.5 UL based on the original protocol. So so it's kind of not economical for us. So we tried scaling down the reactions. So instead of doing the multiplex PCR based on the full volume suggested in the ARTIC protocol, we did it for the half a volume. So that means all of these reagents were scaled down to half. except for the cDNA sample. And we also were able to achieve good results. So basically, scaling down reactions for multiplex PCR will work. So if you don't have many reagents to spare, then you could definitely do this to maximize what you have in your lab. And for recycling conditions, we did not tinker with that. We just followed it as yes. I think we used 63 degrees, but this is based on the ARTIC protocol, because there are other options there in terms of the temperature that you might use. But we stick with 63 degrees. Do you have questions as of now, Stephen?
For the CT value, I understand that you need to test it first, right? It is regardless of any kit that you'll be using, the CT is has the same value.
No, it's not. We don't get the same CT values as reported by the diagnostic labs, because first, the samples have already been stored for quite some time. So there's always a case of a partial degradation. So that affects the concentration of the RNA in the sample. So theoretically, the longer the sample is being stored, the higher the chances are the samples will get degraded through time and have a lower quality or integrity at that. So we just do a retesting just to verify that we can still detect the viral RNA. Otherwise, if we don't detect viral RNA anymore, we just drop the sample totally.
Okay.
But any kit should work. The problem is, for instance, in terms of CT value reporting, different types of qPCR kits will have different performances. So you might have an experience wherein they report very low CT values, but in your kit, you get higher CT values. It's kind of confusing, but we still don't know how to go around that. We just stick with the criteria that as long as we get a less than 30 CT value, then we're good to go.
Okay.
But definitely, if it's just research, you can definitely try to sequence samples with higher CT values than 30, but expect that you will get partially degraded or fragmented genomes.
Okay.
Now, after the PCR enrichment, we don't perform post PCR cleanup and normalization. This is just optional, so we did not do this as well. However, we did two things. The first thing is we ran 1 UL of the amplicon in an agarose gel just to verify that we can see a band, 400 base pair mark. But sometimes at this point, even if you don't see a band at the 400 BP mark, it's kind of a loss if you just drop the sample because you don't have something to replace it. So we just ran the sample along with the batch. But we've seen that generally, if we don't see a clear 400 BP mark amplicon, we get poor sequencing data. So partial genome coverage only. So that means, not all of the genomes were not all parts of the genome were successfully sequenced. So it's best if you get a clear band, because that is an indication that your PCR enrichment for the SARS-CoV-2 genome worked. Another thing is we also measure the concentration of the sample, because the agarose gel is only a qualitative assessment post PCR. We use the qubit fluorometer for a quantitative assessment of the amplicons that we have. And we noticed that not all samples have the same post-PCR efficiency, which means even if you have the same samples with similar CT values, it's not all the time you can expect that they get the same concentration of PCR product. So that could be an issue with variations in like pipetting techniques during your PCR. That could be the case. So what we did was that we made a modification in the dilution process. So in the dilution process, the original protocol recommended that you need only 2.5 UL, sorry, of PCR product per pool. We kind of did adjustments. In the first batch of our run, what we did was for every decreasing band intensity, we added 0.5. So for a very crisp band, we only use 2.5 UL of PCR product per amplicon. And then for a clear band, but not so bright, we use 3 UL. And then faint band, we use 3.5. And then for smear with no band, we use 4 UL. And we were able to get good results from that, but we were not able to sequence successfully everything. Let me share to. Do you see my screen?
Yeah, I can see the spreadsheet.
Yeah. So if you look at this one, this is a qualitative band assessment. So 3+. So if it's very clear and bright. So, but this is subjective. Okay, this is dependent on the analyst. So it's better if we look at the qubit reading. So for instance, if you look at these samples, which we were not able to sequence successfully, we only got very low, like around 30 to 40 PCR amplicons post-enrichment. But yes, we were not able to sequence. 2 samples totally failed. This sample is interesting because, this sample, it has low CT values. You see 24, 24, 22, but it failed in the sequencing process. We were not able to amplify it as seen in this qubit reading. But interestingly, other samples with much higher CT values, like this one, we were able to successfully amplify it. So there's really a variation in terms of, sometimes there are stochastic processes which is difficult to predict and which could affect the samples during the processing. So we always expect that we will get some unexpected results. So like here 26, but still we had higher or better results compared to this one, which has lower CT values, which corresponds to more viral RNA concentration, but we did not get the expected result. Yeah, it's difficult to troubleshoot this because this could be a case of RNA degradation. But when you have an RNA degradation, notice that your diagnostic tests will still pick up the RNA because the markers are very specific, like the S gene, the N gene. So if those segments are still present, then you will have a false impression that lots of viral RNA copies intact are still there, but that's not the case, because if they are degraded, then the rest of the primers used to enrich the viral genome will not work. So that's why you get, that's why these kinds of samples fail. Okay, let me go back to the protocol. Sorry.
May I ask
a question? Go ahead.
Yes. You mentioned earlier when you receive the samples, you check for the quality using cuvette, right?
No. We use real-time PCR.
Okay. So you don't you go direct when you receive the samples, you go directly to RT-PCR first.
Yes.
Okay. How much sample volume do you require for this kind of sequencing protocol?
Okay. For this type of sequencing, you need to have at least 8 UL of starting material. That's per sequencing run. So if you want to do repeats, you need to have more than that. Plus, if you want to retest your sample, you also need to have additional volume to accommodate that repeat real-time PCR.
All right. So
it's safe to say that you need to have at least 20 UL.
At least 20.
But 20. To be sure, 30 would be would give you a lot of
30
room.
UL. Okay. And then do you also try to check the quality of so you go directly to Quebec, because I was thinking of spectral reading to check the quality of RNA, so we don't do
No,
this one?
the spectrophotometer reading is not recommended because what do you call this? It's kind of not as accurate compared to fluorometric based readings. So if you really would want to check RNA concentration, we would recommend fluorometric based assay. There are RNA based assays like qubit RNA based assays, but we don't recommend spectrophotometric reading.
Okay. Okay.
Because
And
they usually the adsorbance that they are measuring is usually more of a blanket general absorbance level, but it does not fully represent the concentration of the RNA that's present. Because it can also measure other things that are double-stranded or single-stranded in the solution, which might not be your target genome.
okay. Okay. And for the gel electrophoresis, have you encountered any primer dimer in the gel?
Yeah, definitely. Primer dimers will occur, but it's not an issue because later on you will do cleanup, and all those low molecular weight amplicons will be flushed out from the system, will be removed.
Okay. 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 That's good.
So that's why there's also no need to do post PCR cleanup. Because we never encounter, we haven't encountered high molecular weight non-specific amplicons, only primer dimers. So they automatically get removed during the cleanup stage because they're less than they're only around 50 base pairs or even lower than that, maybe below 100. But it's really not an issue. But in case if you see bands, higher molecular weight bands, let's say 500 or 1,000. Although we haven't experienced that yet, I think that would entail post-PCR cleanup.
Okay.
But it's quite difficult to do that using the the gel and PCR standard gel and PCR amplification kits, because they only usually remove very low molecular or short segments and very long segments, like the genomic DNA contamination and the primer dimers. So I think you would need to explore another method for that. But so far, we haven't experienced that yet.
Okay. Okay.
So last time we also further modified the PCR dilution process. So based on the qubit reading, so what we did was instead of when you have a qubit reading of below 50, so
that's 50. That's nanogram per microliter.
Yeah, 50 nanograms per microliter. We use 9.5 to 10 UL of the PCR amplicon. If you have a too high, so that's around more than 100 nanograms, we use it should be around like 5 UL of 5 UL microliters of the amplicon. And then maybe if you have around 50 to 100, you can use 7.5. But you can further modify this dilution. Like you can say, if you have too high, you can use 2.5 instead, because if you look at the coverage depth, the 396, that's more than enough for a confident call of the bases. So maybe if it's too high, you can just use 2.5. And then if it's like 75 to 100, you can use 5 UL for the dilution. And then if it's maybe 50 to 75, you can use 7.5. And it's below 50, you can use at least 10 UL or 9.5. Because we don't have enough already after we did the gel and the qubit reading.
How many
flexible.
How many samples are involved in one pool?
In one pool we usually run 23 samples plus a negative control. We haven't. We will try in the next batches to scale up.
I think it depends on the number of what you call this, Micheal? Number of barcodes you have.
Yes. So afterwards, the DNA end repair, we just followed it as is. So 5 UL of your dilution, we subject them to DNA end repair based on this protocol, and then the barcode ligation. Similarly, we followed it as is. Now, for the cleanup of the barcoded amplicons, we use 0.8x of pure XP beads as recommended by the by Ralph from Bort's virology laboratory. They've been doing SARS-CoV-2 sequencing as well, and they noticed that a 0.x ratio of the impure beads to the sample produces better results for the cleanup process. And we also increase the incubation time to have better yield. So the rest is the rest remains as is. So in our first sequencing run, we were able to have around 9.02 nanogram per UL of pooled amplicons. That's following this dilution process for 23 samples. But when we changed the dilution process, we had at least 38.6 nanogram of pooled amplicons. But yeah, we did not retain much after the cleanup.
That's still cuvette reading, right?
This qubit, sorry, not cuvette, qubit.
Qubit.
Yeah, the qubit fluorometer. Okay, for the adapter ligation, as is, the rest of the parameters, we followed it as is. One thing to note is that in the ligation sequencing kit, you only have 2 tubes of SFB. So this is the cleanup buffer.
Mm-hmm.
And for each sequencing batch, you need at least 1 ml. Okay, so one ligation sequencing kit can only support 3 sequencing reactions. So we miscalculated this before. We thought it could support up to 4, but it can only support 3 sequencing reactions for one ligation sequencing kit following the ARTIC protocol. What else did we learn? For the sequencing process, we 14 hours is enough. You don't need to run the sequencing for like 3 days or 24 hours. Because if you have a good input, library input, this time frame is sufficient to have a reliable results.
What time do you usually start your sequencing?
Usually afternoon. So before we started around 3 p.m. The sequencing is already ongoing, and then we just come back to the lab the next day.
Okay.
To clean up the flow cell.
So you just leave it sequencing overnight.
Yeah, and we leave the aircon on because we need to maintain a particular temperature conducive for the machine. In terms of flow cells, so flow cells, we're thinking right now that we can only reuse a flow cell once. After the first reuse, the flow cell, so far, we tried reusing a 1 flow cell so far. And then after the first or the second run, the flow cell is already exhausted, so you cannot use it for additional run. And our previous run last week, or was it 2 weeks ago, we used a new flow cell. We washed it And then after having a flow cell check, we only recovered like a few hundred pores, like around 200 to 300 pores. So we think that that can only be used once more, and afterwards it will be exhausted. So some other people are saying that you can reuse flow cells up to 4 times, but that's very optimistic. It doesn't look that we can do that for this one. And when we I think it would be best if you immediately use that for sequencing, because the longer you store that, the more chances of the pore quality decreasing, so you won't have enough pores anymore. What else can we share? Do you have any specific questions, Stephen, in terms of the process?
I would probably want to ask how many lab personnel is involved in executing all these protocols.
Okay. So I think 2 personnel is sufficient, 1 lead and 1 assistant.
And how long is it to execute all this up to generating the sequence?
So in our experience, we allot 1 day for the enrichment. That means from cDNA synthesis to multiplex PCR and the gel and qubit quantification. So this one, from part B to part C, we allot 1 day for this. So when we do retesting, we do it another like a day before or
a day before. Okay.
Yeah. So here, this is really the start of the library prep process from the cDNA synthesis, and after here, you can even do the dilution already. You can do this in 1 day, from the cDNA synthesis to PCR dilution, and we leave we start the DNA end repair the next day already until the sequencing. So in day 2, when we start at 9 for the DNA end repair, we can start the sequencing up to this step at around 3 p.m. So that's like 5 hours, because you still have to take a 1-hour lunch break.
Okay. So you mentioned that you let the nanopore sequence for 14 hours. Is it correct?
Yeah.
How do you know when to stop the sequencing?
It will automatically stop once the 14 hours.
So it's preset to 14 hours, then it stops.
Yes. So here, before you start the sequencing, you need to specify the settings. So you need to specify the kit, and then you set the run time. So 14 hours. We turned off the base calling option, so that means the sequencing will only produce the raw files. There's an option wherein it can do a real-time or live base calling, which means that as the data comes in, it's being converted into sequences, but it takes a higher computational resource to do that. And we were using right now a desktop computer with an 8 GB RAM, so we did not think that would be suitable for that resource requirement. So we can just turn it off. So the sequencing will produce the raw fast file, and then the bioinformaticians will process it downstream to produce the sequencing output.
So after it has finished the sequencing, how do you process the sequence data?
I think it's the bioinformaticians that does that. They use softwares wherein they tinker with the data, and it produces the results that you're looking. But I think Paul would be the best person to run down the protocol. He's our bioinformatician at PGC Mindanao.
Okay.
Another thing, here in the benign sequencing, you need to be familiar with loading the library, and some steps might be confusing. So I suggest that you watch the tutorial videos for nanopore sequencing. It's freely available on YouTube. So they have videos in how you prepare your flow cell, what you do, how you properly load the library, and so on. And
That's
very informative.
That's a Nanopore video.
Yeah, they have it on their
Okay.
YouTube channel.
Okay. I will check on that.
Because sometimes the terms are confusing, like, priming port, they say here, inlet port, priming port. So yeah, these terminologies might be confusing at first, but they're just referring to some parts of the machine. So it would be best if you watch the video.
Although I would suggest that why you don't you should make the video yourself.
Sorry.
I think it would be on my side. Why don't you try to record when you perform the sequencing, all the steps?
Yeah,
it's
actually,
like video tutorial.
Yeah, we had someone cover our last sequencing run, but I do know that making such videos are really time consuming. So
Okay.
I don't know with the promotions team.
I think I have no questions anymore at this point. Is it okay to have a copy of your protocol?
Yeah, definitely. We can share that to you.
All right, thanks.
That's largely based on the ARTIC protocol. We only did minor changes based on our consultations with other laboratories.
Well, I think the most significant modifications you did was when you reduce the number of reagents in one of those steps. You optimize
the scaling down.
Yes, the scaling down. I think that's very applicable to us here in the Philippines.
Definitely, we've been doing that for in PGC Mindanao. I have a lot of experience of scaling down reactions just because of the idea that we don't have enough. And so far, yeah, in theory it should work as long as you do it proportionally and the final concentrations are relative to each other are maintained.
Okay, so may I know how many times have you attempted the sequencing part before you were able to get good data out of it, or just the first time you did it and then you have a good data?
Yeah, the first time we did it, we already have a good data.
Although
we tried the really first run, or the very first run that we did, we used a wastewater sample. So that's a poor quality sample. The CT values are very high, like 33 to 40, 39. So that's a very fragmented and degraded viral RNA in the sample. So the results, as expected, were not good because of the nature of the samples.
Yes,
but if you're using high quality samples, you can expect that the first time you do the sequencing, as long as everything goes right, you will get relatively good data.
Okay.
It might not be perfect. It will not always be perfect.
That's correct. All right. So far, I have all my questions answered. What else do I need?
Mitch, I have
okay, one more.
Go ahead, Stephen.
One more thing is on the biosafety protocol side. What are the important biosafety protocol are you practicing when performing the sequencing?
Okay. In our lab, we do not accept live specimens. So we only accept extracted viral RNA. So these are non-infectious material. So basically, it can be processed in a BSL-1 lab or in a general lab. You don't need a biosafety cabinet to do the sequencing because, as I've said, it's non-infectious. It's not pathogenic anymore, so it's quite stable.
So when you prepare your library, you don't have to use any biosafety cabinet. You just do it on the bench.
When we do the PCR, we for the sake of reducing the likelihood of contamination, also for the reagent's sake, we do the PCR preparation in a PCR hood.
Okay.
Yeah. And
So
then the
essentially,
library preparation can just be done on the bench.
okay, so that's biosafety level 1. Okay.
Yes.
Okay, I understand.
But that's a different story if you will be starting from live samples. So you need to have a BSL2 plus lab to do the extraction.
And then another thing is, do you need to have like a permission from any authority when you do sequencing?
Tough question. If you want to publish, you need to have an ethics approval.
Okay, it takes approval.
Yeah, but currently, as far as I know, there's no regulatory body for doing genomic sequencing. So if it's just for internal purposes like your own surveillance, you can definitely do that. But if you want to publish your output, you need to have an ethics approval.
Okay.
Stephen,
And then, yes,
Stephen,
yes.
regarding that, you might want to reach out to Kirsten as well. So we partnered with her because they already have ethics approval. So they just added PGC Mindanao plus the partner labs here to their ethics approval as an amendment. So that might, if you
I
want to
see.
publish, you could just approach her and ask if you could be added as well as a partner lab.
I see. Okay.
You would also have help in terms of bioinformatics if you partner with them because they have good bioinformaticians who could help you.
Ethics approval. Okay. All right. So I think I have everything covered. That's one of the main issues I want to ask actually is the biosafety protocol. Although Christine also connected me to one of the personnel at RITM, but I want to know also on your side what protocols are you observing, especially on the biosafety side, because
Yeah.
we know that when we say we want to sequence our uh, uh, for COVID samples, then most people who don't understand will definitely say, no, it's it's infectious. So at least I have something to cite. Oh, no, they're doing this, so it's BSN level
If you're
1.
only handling a viral RNA, that should be okay because it's non-infectious.
Yes.
I think you might also want to seek approval from your institutional biosafety and biosecurity committee if you have one at your institution.
Okay. On your end, how, how long did you wait for the biosafety approval?
For the biosafety approval,
I mean for the ethics. I mean for the ethics approval.
For the ethics, I think
Yes.
Khalil can answer that.
Yeah, so there were several attempts, Stephen. So the first attempt was with DOH in one region in Mindanao, and then that never pushed through. We waited for months for that. That's why we approached the Gecko project and asked if we could be part of it and then be part of their ethics approval as well. So that one, I think it took a month from submitting the amendment until the actual approval. So it will take time to wait for the next batch of like amendments, plus maybe another month for the actual approval.
I see. But you mentioned that for publication purpose, you need ethics approval. So if, for example, on my part, I don't want to publish, I just want to say sequence it. Do I still need to have this? I think so. Yeah, because it's,
well, there's I could send you a document regarding that. It is not clear. So okay, I think so. The most strict, what you call this, the guidelines in the Philippines is most strict when it comes to human samples or sequencing human DNA. But in this case, this is a viral RNA sequencing. It's there seems to be no regulation in terms of that. This is just being what you call this, safe in terms of, actually it's not clear. Up till now, it's not clear what are the regulations. We're just following what what's being asked. So what the others are doing.
Well, on my end, we have a university hospital, and they have their own ethics committee. So I was thinking if I'm going to do this sequencing, I might try to apply for ethics approval from our own institute. I'll just check on how long will they
I
process
think
it.
that would be enough.
That will be enough now.
Yeah.
Okay, okay. I'll try
As
to do
long as
that
you have your
first.
institutional approval,
Mhm.
since you are a medical, you're from a medical institution, you have a yeah.
Yes, I'll try to do it first. Maybe I can get a faster ethics approval.
Yeah, a key, maybe an important detail there, Stephen, is the what you call this, the document that the patient signs. What you call the thing? So
The informed consent.
informed consent, because to be safer, it should include like some language which informs them that it's not only PCR that will be used to read their
Okay,
data,
okay.
but it could be like a
Okay.
more general language.
I see, I understand. So it does not necessarily mean that if the patient has approved the testing, the RT-PCR testing for COVID, already includes the sequencing. So I need to have that consent also from them. Okay, I understand.
Ideally, I think that would be for the best, but there's really no clear guidelines on that. As far as we know, we are also not. I think some other hospitals are doing post, like they're getting the consent after,
Okay.
but that would be tricky.
Okay. But
I think in UK, as we know, based from Kirsten, these samples are considered residual diagnostic samples, so you don't need to get any prior informed consent because you're not dealing with any patient metadata and other privacy concerns, or you're not like referring them to any identifiable individual. So, but yeah, the guidelines are not consistent across regions or even countries. Yeah, countries.
I think at this point it would be on our advantage that there's no yet strict regulation. So I might not well try to ask for it. Okay. So
You could also talk to
sample.
Kirsten, Stephen, regarding, because I think they have the existing format for the informed consent, because like your existing informed consent might already fit that. I mean, if it's not too specific, meaning this is a sample and we will use your sample for studies, and I consent to that. I mean, what's the difference between an RT-PCR versus sequencing? It's really just the level of what you call this specificity of like the information being extracted from the sample, and also the sample is not the human sample. It's like the virus that happened to be in a human being.
Yeah.
And what data do you get from the samples coming in from you? Is it only the sample number and nothing else? No information about the data?
For research, we need to use the metadata, the clinical metadata for comprehensive assessment of results. So since we are using clinical data, we need to have a really stringent ethics approval because for publications purposes.
Okay.
But if your purpose is just general surveillance, you just want to know the proportion of samples in your area which can be classified as such as particular variants, then you won't be needing those metadata. But to be fair, it will be advantageous if you can also interpret your sequencing results along with your clinical metadata. So that's where ethics comes in.
Okay. All right.
Mitch, I have a question. So
yes,
we have so currently what we're purchasing is the SLK 109 starter kit plus the NEB Next kit. So what are the reagents in the ONT starter kit, Minayon starter kit, which you need to supplement when you buy it based on the usage that you've experienced?
Sorry. Okay. You need to buy a barcode kit. So yeah, the barcode kit will allow you to multiplex samples for your sequencing run, depending on the number of barcodes that you have. So for instance, if you only have like the 1 to 12 and the 13 to 24 barcodes, you can only multiplex up to 24 samples per batch. So if you want to go higher, you need to buy the 96 barcode kit, wherein you can run up to 96 samples per batch. And for each barcode, you can use that up to 6 reactions. So that means if you buy a 96 barcode kit, that's good for 96 times 6 reactions. And additionally, you also need to buy flow cell wash kit because if you want to reuse your flow cells to salvage the remaining pores and utilize it for another sequencing run, you would need to wash your flow cells after each sequencing.
Mitch, you mentioned
after sequencing.
So, there's a wash kit, right, in the starter kit?
Sorry. Yeah. The wash kit in the starter kit is sufficient. It's actually the SFB.
That's a buffer, ligation buffer.
The short fragment buffer.
Okay.
One ligation sequencing kit can only support the SFB can only support 3 ARTIC sequencing runs. If you want to, but based also on our experience, wherein we can only reuse one flow cell, the flow cell once, that would be enough.
Okay, but Mitch, we ran out of that, right?
Yeah.
So meaning for every starter kit we purchase, we should add another one more vial of that, or one more batch of that.
Okay, assuming that for each starter kit you have 2 flow cells, you can run up to 4, a minimum of 4 batch of samples. So if you have if you want to sequence 4 batches of samples, the SFB in one ligation sequencing kit is not enough. So you need to have an extra. Supplemental SFB kit. But the supplemental SFB kit, I'm not sure about how many washes can it support.
Okay. So that's the only one. That's the only one which needs to be supplemented.
Yeah.
Okay. All right. Any final words, guys, before we end this call?
Do we have a timeline for this one, like the reagents part?
Yeah, we're still waiting, Micheal, for the delivery of the new ones.
Yeah.
Okay.
So it was supposed to be today, but they messed up on their side.
All right.
And we will just update you, Stephen, just about that at least we know what's already there. Mitch, we could also give a bit of some of the would we spare some of the barcodes?
Barcodes. Yeah, we can definitely send barcodes to them as well.
Cool.
All right, that's great.
We have more than enough.
All right, and then the 96 kit barcode is coming, so that we should have sufficient. All right. So if nothing else, thank you so much, Mitch and Stephen, for this call.
Thank you.
And good luck with the sequencing.
Thank you.
Thank you. Good luck, Stephen.
Thank you.
All right, goodbye.
Bye.